Hi everyone, welcome back to the Science Box. So today I'm going to be telling you everything you need to know for the Cell Recognition and Immune System chapter of AQA A Level Biology. The timeline's in the description, so if there's a particular part you're stuck on, feel free to just skip to there. I'm going to be starting with what a lot of this chapter revolves around, which is antigens. An antigen is any part of an organism, usually proteins on the cell surface membrane, that is recognised as non-self by the immune system and stimulates an immune response. The initial immune response stimulated is phagocytosis. Okay, so step 1 of phagocytosis, chemical products of the pathogen act as attractants, causing phagocytes to move towards the pathogen. The phagocytes have receptors on their cell surface membrane that recognise and attach to the chemicals on the surface of the pathogens. They engulf the pathogen to form a vesicle known as a phagosome. Lysosomes move towards the vesicle and fuse with it, and then enzymes within the lysosomes, called lysozymes, destroy ingested bacteria by hydrolysis of their cell walls. The soluble products from the breakdown of the pathogen are absorbed into the cytoplasm of the phagocyte. You can remember the gist of this through the acronym DED, D-E-D, Detect, Engulf, Digest. Phagocytes detect antigens on the surface of a pathogen as non-self, engulf the pathogen, and then digest it, destroying it. Phagocytosis is non-specific, but the rest of the immune response is specific. Now moving on to T cells. Both T cells and B cells are a type of white blood cell called a lymphocyte. All lymphocytes are produced by stem cells in the bone marrow. T helper cells are stimulated by antigen-presenting phagocytes called macrophages, that present some of the pathogen's antigens on their own cell surface membrane. The T lymphocyte only responds to the antigens that are presented on a body cell, rather than just antigens free in the bloodstream. So that's why this is called the cellular response or the cell-mediated response. Receptors on a specific T helper cell fit onto these antigens because they're complementary. This attachment activates the T cell to divide rapidly by mitosis and form a clone of genetically identical cells. The cloned T helper cells activate cytotoxic T cells. Cytotoxic T cells kill abnormal cells and body cells that are infected by pathogens by producing a protein called perforin that makes holes in the cell surface membrane. The cloned T helper cells also stimulate phagocytes to engulf pathogens by phagocytosis, to develop memory T cells that enable a rapid response to future infections by the same pathogen, and they stimulate B cells to divide and secrete their antibody. This brings us on to the response that's all about B cells, the humoral response. The B cell with the complementary antibodies to the antigen binds to the antigen to form an antigen-antibody complex. The antigen enters the B cell by endocytosis and gets presented on its surface, and this is called processing. The T helper cell binds to these processed antigens and stimulates the B cell to divide by mitosis to form a clone of identical B cells. This is called clonal selection. This clone of cells could either be plasma cells or memory cells. Plasma cells secrete multiple antibodies into the blood plasma but have a short lifespan. The antibodies combine pathogens together in a process called agglutination to allow for quicker phagocytosis and act as markers that stimulate phagocytes to engulf the pathogens. This is a primary immune response. Memory cells are responsible for the secondary immune response. If they encounter the same antigen at a later date, they divide rapidly and develop into plasma cells and more memory cells. So an increased quantity of antibodies is secreted at a faster rate than in the primary immune response. So memory cells provide long-term immunity against original infection. Now let's have a closer look at antibodies. An antibody is a protein produced by lymphocytes in response to the presence of the appropriate antigen. Antibodies are made of 4 polypeptide chains, so you can see the 2 purple ones on the inside and the 2 pink ones on the outside, and each antibody has 2 identical binding sites which are complementary to a specific antigen. A lot of people think that the antigen fits into the gap between the binding sites, but every antibody actually has 2 binding sites on the end of it for the antigens. Monoclonal antibodies are antibodies produced by a single clone of cells. One of the main uses of monoclonal antibodies is targeting medication to specific cell types by attaching a drug to an antibody. So the antibody travels through the blood and binds to the antigen on the target cell, bringing the drug with it. This is used on cancer cells where monoclonal antibodies are produced specific to the antigens on the cancer cells. Then these antibodies are given to patients and attach themselves to receptors on the surface of the cancer cells. Once attached, they can then block the chemical signals that stimulate the cancer cell's uncontrolled growth. Or, in some cases, a radioactive or cytotoxic drug is attached to the monoclonal antibody so that when it attaches to the cancer cell, it just kills it. Monoclonal antibodies are also used for medical diagnosis to detect the level of antigen in the blood. And they're also used for pregnancy tests. Let's look at how this works in more detail. So on the left we have what happens when someone is pregnant, and on the right we have when someone isn't pregnant. When someone is pregnant, the hormone HCG is present in their urine. The urine travels up the strip and first reaches the reaction zone. Here there are mobile antibodies with blue dye attached to them that are complementary to the HCG hormone. So on the pregnant side, you can see they bind to the hormone, and then on the other side, the hormone isn't present, so they can't bind to anything. The urine then travels up to the results window. Here, there are immobilized antibodies that bind to another binding site of HCG, and the dye releases when this complex forms. This is why on the pregnant side, which has HCG, there's a blue line in the results window, and on the non-pregnant side with no HCG, there's no blue line. The urine then travels up to the control window, which has immobilized antibodies specific to the mobile antibodies that started in the reaction zone. Both tests have these mobile antibodies that should travel up the strip, so a blue line should appear in both control windows. This verifies that the urine travelled all the way up the test strip. So yeah, that's the whole process of a pregnancy test, but feel free to go over that again because it is quite complicated. We can also use antibodies to do an ELISA test, which is a test we can do to detect the presence and quantity of a protein. Step 1. Apply the patient's sample to a surface, like a slide, to which all the antigens in the sample will attach. Wash the surface several times to remove any unattached antigens, and then add the antibody that is specific to the antigen we are trying to detect, and leave the two to bind together. Wash the surface to remove excess antibody, and add a second antibody that binds with the first antibody. This second antibody has an enzyme attached to it. Add the colourless substrate of the enzyme, and the enzyme acts on the substrate to change it into a coloured product. The amount of antigen present is relative to the intensity of the colour that develops. Although monoclonal antibodies have multiple uses, there are ethical issues with them. The production of monoclonal antibodies involves the use of mice and deliberately inducing them with cancer. Monoclonal antibodies have been used successfully to treat diseases, but also unsuccessfully and resulted in death, so patients need to be informed about the full risks and benefits. And also, testing for the safety of new drugs comes with its dangers, so healthy volunteers can be killed. Now moving on to vaccination. There are two types of immunity, passive immunity and active immunity. Passive immunity is produced by the introduction of antibodies into individuals from an outside source. Artificial passive immunity happens by the injection from a donor, and natural passive immunity can happen, for example, from mother to child. Passive immunity is short-lasting, but the immunity is acquired quickly. Active immunity is produced by stimulating the production of antibodies by the individual's own immune system. Natural active immunity is getting the disease under normal circumstances, and artificial active immunity is vaccines, because a dead or inactive form of the pathogen is injected inside of you. From active immunity can come herd immunity, which is when a sufficiently large enough proportion of the population has been vaccinated to make it difficult for a pathogen to spread within that population. Pathogens can mutate so that the shape of the antigen changes suddenly, and this leads to antigenic variability. The ethical issues of vaccines are that the development of vaccines often involves animal testing. Vaccines may have side effects, so you have to deliberate between the side effects and the risks of catching the disease. There is a question of should vaccines be tested on healthy volunteers, and should vaccination be compulsory. We're now going to look at one disease in particular. It's a virus, and the virus is HIV. HIV is a type of virus called a retrovirus because it contains a type of enzyme called reverse transcriptase, meaning it can make DNA from RNA. Here's a label diagram of a HIV particle. This is really similar to the diagram of a generic viral particle we labelled in the cell structure video, except we've specified the genetic material as RNA because we know it's always going to be RNA. So, knowing this structure, we can now look at how HIV integrates itself into the host. Following infection, HIV enters the bloodstream and circulates around the body. Attachment proteins on HIV readily bind to a protein which is most commonly found on the T helper cells. The protein capsid then fuses with the cell surface membrane of the host, and the RNA and enzymes of HIV enter the T helper cell. HIV reverse transcriptase then converts the virus's RNA to DNA. The newly made DNA is moved into the T helper cell's nucleus, where it's inserted into the cell's DNA. The HIV DNA in the nucleus then creates mRNA using the host cell's enzymes. This mRNA contains the instructions for making new viral proteins and the RNA to go into the new HIV particle. The mRNA passes out of the nucleus through a nuclear pore and uses the cell's protein synthesis mechanisms to make HIV particles. The HIV particles then break away from the T helper cell with a piece of its cell surface membrane surrounding them, which forms the lipid envelope. HIV attacks helper T cells, reducing the number of them. Therefore, B cells and cytotoxic T cells can't be stimulated. This leads to inadequate immune response, so the patient becomes susceptible to other infections. One way of treating diseases is using antibiotics, but antibiotics only work on bacteria. They weaken the murine cell walls by inhibiting certain enzymes required to make it, so the cell bursts and the bacteria die. Viruses have protein coats and not cell walls, so there's nowhere for the antibiotics to work. And that brings us to the end of the cell recognition and immune system chapter of AQA A Level Biology. Thank you for watching. Please like and subscribe. If you like this style of teaching and would like to get tutored by me or a member of our team, you can go to our tutoring website at thesciencebox.net. I'd also really recommend our Instagram and TikTok for tiny doses of revision. It makes you feel productive even if you're just scrolling TikTok, so that's a bonus.